All right, we're going to do the final little piece of our journey here. Jack has his manuscript in Italian, and he's going to take it to the translator, and the translator is going to do some magic, going to work some magic, and he's going to give Jack the same set of information, but in English instead. To translate that back to our biology example, we have our piece of mRNA here. I didn't show the modifications, but assume there's a 5' cap right there. It would just take up too much space and get in the way. There's a poly A tail right there. Again, it would take up too much space to get in the way. And I've removed all of the introns that I wanted to remove, and I've stitched together the exons. So this is now our ready-to-go mRNA. We're going to do some work on this mRNA, or rather an object in the cell is going to do some work on this mRNA, and it's going to produce a sequence of amino acids for us that we're eventually going to modify and call a protein. And the object in the cell that's going to do that is an object that in this little demonstration looks kind of like this, and if you look at some images online and in your textbook you'll see it looks sort of similar. This is the ribosome. The ribosome is the organelle in the cell that makes protein. Hopefully you remember that from grade 10 science, I think from grade 7 science as well. And the ribosome has a large subunit and a small subunit, and some magic happens inside the ribosome. The ribosome doesn't actually physically do the translating itself. It provides the location for the translation to happen and also checks to make sure the translation is done properly. Ribosome is made up of a type of RNA that we call rRNA, or ribosomal RNA. So that's the second of the 3 pieces of RNA that we're going to use in this process. The third of the 3 pieces of RNA that we're going to use in this process, it's unfortunate my color printer ran out, so I'm working with this. This is in the document that I posted on D2L, and you also have in your course notes. This is in the translation document. This is a molecule called tRNA. So maybe you want to pause the video now, have this document in front of you so that you can see it. You can refer to it as I'm talking about it. tRNA is called tRNA, or rather the T in tRNA stands for transfer. It's transfer RNA. Transfer RNA also is kind of shaped like a T, right? So it pulls double duty there. Transfer RNA is the molecule that's actually going to bring the amino acids to the ribosome, and it's going to give us the proper amino acid in the proper place along the code of our mRNA. So I'm going to push this off to the side. We'll talk about it in a little bit. We'll get back to it. For now, we're going to work with our process. So we have a 5' cap. There are some signals at the 5' cap that the ribosome is attracted to. So the ribosome comes on to the 5' cap, starts there, and then very slowly, in reality it's very quick, but for our purposes it's very slow. The ribosome is going to move along this piece of mRNA, and it's looking for a sequence called AUG. AUG is what we call our start codon. AUG is always, for our purposes here, where we start the process of translation. And the word codon, which is, it might be a new word for you if you haven't watched any of the other videos or done any of the other readings, the word codon is 3 nucleotide code. So AUG, 3 nucleotides in a row, that's a codon, and then we have other codons here. So once it finds AUG, what it's going to do is it's going to allow the tRNA that has the opposite or the anticodon to AUG, it's going to allow that tRNA to come in. So in the handout here, in the handout that I posted and that you have in your course notes, the complementary codon to AUG, it would be UAC. In the handout, the anticodon listed here, which is the same complementary, is GAA. So this is not the one that we're working with right here. This is a different tRNA, but there is a tRNA that has this as the anticodon. It has UAC. And UAC, that tRNA, would come in and it would bring with it an amino acid. And the amino acid that always is brought in by that piece of tRNA is the amino acid methionine, MET. Then, once that methionine is in, the ribosome is going to continue on with what it was doing. The ribosome is going to move. It's going to slowly move its way down, and it's going to go to the next codon. So we can start blocking off our codons here. We have AUG, that's a codon. And then every 3 bases after that is another codon. So we're at AAA. So an amino acid is going to be brought in by a tRNA, and the amino acid for AAA is the amino acid lysine. So we're going to get an amino acid put in place called lysine. And this connection right here is a connection that you've already learned. We learned it, I think, during week 1 of distance learning. This is a peptide bond. The connection between 2 amino acids is made within the ribosome. It is our peptide bond that we learned earlier. So we have 2 amino acids in a row. We have a dipeptide. The ribosome is going to keep moving. It's going to go to GUG. And the amino acid for GUG is valine. So there's my amino acid. It's VAL. And I have another peptide bond. And now I have a tripeptide. And eventually I'm going to start calling this a polypeptide. I can, tetrapeptide, pentapeptide, just polypeptide from this point on. The ribosome moves down, GGC. Now, you might be wondering, how does Mr. Vona know? How did he memorize? Did he memorize which amino acid goes in which place? So we're looking at GGC. GGC is glycine, GLY. And I did not memorize it, and I don't expect you to memorize it, and I hope none of your teachers going forward expect you to memorize it. This is another page from your course notes. I'm using this. I'm sorry I can't produce a bigger image than that. You have this in your textbook. You have it in your course notes. It was in the document that I posted. This is the code of life, or what we call the genetic code. This is your Italian to English dictionary. This is what tells us what amino acid will be put in place for which codon. So you have the first letter of the codon on this axis, then the second letter up here, the third letter written in there, and you would just find, if I'm looking for AUG, I would find A. There's U right here down this column. So in this box, AUG, MET, methionine. And I know that when I have AUG, I have methionine. And AUG is always going to be our first codon. AUG we call the start codon. And that means that every single protein that you guys have to make starts with AUG. And it always starts with methionine. The process will keep going. So our next is UCC. Take a look at your code of life. If you don't have it in front of you, pause the video, go get it. Take a look, UCC. As I'm drawing here, hopefully you found SER, serine. And then the process keeps going. The ribosome moves again. We get UAG. And we have what we call a stop codon. Now, a stop codon isn't actually a codon. There's no amino acid, or it is a codon, but it doesn't code for an amino acid. So this amino acid has to go. So what we have in our amino acid chain is only 5. And UAG, our stop here, tells the ribosome that this is where the protein is done. So we have extra bases at the end. That doesn't make a big deal. They're there, they just hang out. They don't really get involved in the process. We have extra bases at the start. Notice that I didn't start with AC. I didn't start ACA as my codon because I'm always looking for AUG. Every time there is an AUG, a tRNA is going to come in that has the anticodon, and that tRNA will always have methionine. Every time I have GUG, the tRNA comes in, will always have valine, always. If there's ever a mistake in this type of translation, like you have a mutation in you that changes which tRNA carries valine, which tRNA carries glycine, that is almost definitely going to be something that kills that organism in the first few hours of life. This is responsible for making every single protein, and you can imagine that 1 out of every 20 amino acids would be wrong. That's a 5% change in the proteins. That's a huge change, and these proteins wouldn't be able to survive. So we have our codon, 3 nucleotides. We always start with AUG, that's our start codon. If you take a look at the code of life, you'll see that there are 3 stop codons. They don't actually code for an amino acid. The process is just done. tRNA is our translating device, and it also stands for transfer RNA. rRNA makes our ribosome. mRNA is our ready to go piece. And now we have a protein here, and this protein is ready to get to work within the cell. We have a useful object from the information that was interesting and important, but didn't actually do anything. And that's translation.